Brain aging is a huge problem.
In the heart of Northern California, a revolution is underway that could redefine the way we age.
We came across a difference between the male and the female brain during aging that really took us by surprise.
Dr. Ashley Webb's research is uncovering the secrets that keep our brains youthful.
When you go into the lab, that's true discovery, right? You're asking a question and you're designing an experiment, and at the end of the day, you get an answer.
Méline is the founder of the HumanWare project. Driven by a deep curiosity, she's traveling the world to uncover the hidden secrets shaping the future of our health. Today, we step inside Ashley's lab to explore the groundbreaking discoveries she's making. With mounting evidence that men and women age in different ways, her goal isn't just to slow down aging, but to make interventions targeted and truly inclusive for everyone.
The brain can actually regenerate itself to some extent. Breakthroughs are coming very quickly in that area. And the potential, I think it's going to help a lot of people.
The Buck Institute was established in 1999 and designed by renowned architect I.M. Pei. It's the world's first independent biomedical research institute solely dedicated to aging and age-related diseases. Dr. Webb's research spans everything from memory loss to sleep disruption, all with the goal of helping us stay healthier as we age.
So we are at the Buck Institute for Research on Aging, and we are in Novato, California. To me, it's a very inspiring place to do science. Fortunately, up here, we are right in nature, and that's one of the benefits of being up here. We have lots of deer, wild turkeys, and other species that come by as well. It's very, very cool.
That's beautiful.
So welcome to the lab here at the Buck Institute.
It looks like a very interesting lab. They have so many things going on here.
So this is where we do all of our molecular work, studying brain aging, neurodegeneration. One thing that we can do in the lab is we can actually generate human neurons using reprogrammed cells that were actually initially obtained from patients. And so once we have these neurons in a dish, we can actually compare the disease cells to the healthy cells to begin to understand some of the molecular processes that are going wrong in the disease cells to eventually find targets to try to improve the health of those cells.
And how long is the process going to take?
Actually takes weeks.
Ooh.
Yeah.
Didn't expect to be so long.
The great thing though is that then what we have is neurons that are fairly mature, so they actually have features of Alzheimer's disease that we can study and then compare them to the healthy cells to discover molecular targets. One thing you always need in the lab is centrifuges, and it's critical for tissue collection or isolation of parts of the cell. And then this would be a rotor that would go into that centrifuge, for example, and it's actually very heavy and it spins very fast. So tens of thousands of G in order to collect samples. Proteins, organelles, RNA, DNA, those can all be isolated using the right solutions in order to collect them at the bottoms of the tubes. The great thing is that once we collect tissue, we can keep it for a very long time and everything is tracked and cataloged, and so we can go back and analyze things over time. So we actually have some tissue that we've obtained from brain banks that was collected in the 80s, in the 1980s. So years and years. If it's stored properly, it will actually still be good. All right, well, I'm excited to show you some cells. That's where we keep our cells. And we're going to need some safety equipment for this one.
Thank you.
So the great thing is that once we have cells that are in culture that we're growing and studying, we can actually freeze them down. And that's a good thing because if we need to go back to them later, a year later, 2 years later, we can actually go back to this liquid nitrogen tank where the cells have been frozen and bring them out and put them back into culture and then study them all over again. And so this is the liquid nitrogen tank. Very cold. Right now it's about negative 150 degrees Celsius. If it splashes in your eye or anything like that, that would be a very bad thing. And so this tank is basically full of racks and racks of cells. So for example, we can pull this out. In this box, you can see there are vials of cells. It's very cool. All right, pretty cool.
The lab offers a glimpse into the complexity of aging. But to truly understand where the science is headed, Méline joins Dr. Webb for a deeper conversation about what's driving her work and the potential it holds for all of us.
Brain aging is a huge problem, right? So in our society, we know that with age, there is clearly cognitive decline, and that actually happens as part of normal aging. So, that's something that we currently are not getting around. Yeah. But we also know that the number of individuals who are suffering from dementia-related diseases, so things like Alzheimer's disease, is increasing. And so we need to pay attention to our brain health, not only to maintain our cognitive function, but also to keep our brain healthy and disease-free with age.
Do you think from early stage, if we pay attention to our brain health or neural health, that decline of cognitive health can be avoided?
Absolutely, yes. Generally in the public, we're starting to gain this appreciation that there are things that we can do early on, right, to prevent disease and to ensure longevity. You don't want to have brain injuries early in life, right? So that's an extreme example, and we know that things like concussions early in life, traumatic brain injury, can actually lead to even earlier onset dementias, and that's a very scary thing. So that's that's one thing, but that's not happening to most people. Here we're talking more about things like diet, exercise, things that we actually can control in our lives. Be active, eat well, don't smoke, don't consume too much alcohol, and so there are these simple lifestyle interventions that we actually have control over, and that's the good news, I think.
Could you break us down what each part of our brain do, and what is your lab particularly focused on study, and what are the results?
So this is a great question. So the brain is obviously a highly complex organ with a number of different brain regions, but they're also highly interconnected, of course. And we are focused on two main areas of the brain. One is known as the hippocampus. The hippocampus is a critical center for learning and memory. And unfortunately, we know that the hippocampus is actually one of the most affected areas in Alzheimer's disease. And so we've become particularly interested in that part of the brain and how to keep it healthy throughout life, and then what exactly happens to it in the context of aging and Alzheimer's disease, with the idea that if we can identify targets, molecular targets that is, that are changing with age in this part of the brain, then we can go in and come up with strategies to improve the function of the hippocampus with age.
For a person who have a very highly efficient function of hippocampus versus the people who start to experience decline or they have really dysfunction ones, what the difference?
The hippocampus is critical for the formation of new memories. So it's not an area of the brain where memories are actually stored long term, but you need the hippocampus for the processing of those memories and for the retrieval of those memories. And so when we think of things on a daily basis where we're just generally forgetful, a great example, and it relates to the work that we do specifically on neurogenesis, is actually finding your car. So you go to work in the morning, right? You drive your car, you park it in a particular spot, you work all day, and then you go back out, and where is my car, right? So the young brain is actually very good at that task, but the older we get, the harder it is to actually go back out and find your car. And that sounds like a small thing, but if you think about tasks like that, we're doing those tasks all the time. And if you start to lose that function and it erodes over time, it's actually quite disruptive for daily life.
When I travel by myself, I actually going to pay more attention. Would you say that is when my hippocampus is actually engaging?
Multitasking, we know, is something that the brain just doesn't do very well. We think that we can multitask very well and use our hippocampus to keep track of the road, and then use another part of our brain to engage our friend. But in fact, neither one of those is actually going as well as you think it is at that particular time. And so you're absolutely right. And with age, it's even harder to set that map and then remember it.
Hippocampus, what their job is to bookmark what we see during the day And probably when we sleep, they're going to restore into a longer form of memories?
That's absolutely correct. So this is one reason why we need sleep. We need sleep for a number of reasons, but memory consolidation is one of those things. So during the day, our brain is very active, it's jumping from task to task, and then at night, we need healthy sleep in order to consolidate those memories for the longer term, and so that's long-term memory. And that's another thing that is eroded with age, in part due to issues that we have with sleep during aging.
If our hippocampus or if our bookmark system is getting broken, is there any interventions or things we could do when we start to feel that way?
Absolutely. And a lot of this, again, goes back to, straightforward lifestyle interventions. So, going back to this idea of healthy sleep, that is one of the best things we can do for our brains. And unfortunately, like I mentioned, with age, it's one of the harder things to stay on top of. Simple things like making sure the room is cool at night, that we have absolute darkness, sleep with an eye mask if you need to, go to bed at the same time every night, turn off your devices, things that we consider sort of sleep hygiene can really go a long way towards promoting healthy sleep.
From a neuroscientist perspective, could you explain in a very simple form what happened to our memory, to our brain while we sleep?
The brain, it doesn't turn off during sleep, right? So the brain switches into a sleep state and there is a firing of neurons still during sleep, and it changes when you are in REM sleep versus non-REM sleep. But that firing that occurs during sleep is critical for consolidating those memories.
Is man's brain different with female's brain, and are we aging the same speed?
So this is a fascinating question for me. A couple of years ago, we came across a difference between the male and the female brain during aging that really took us by surprise. We got into this work in part because we know that with aging, there is a bias when it comes to how women age versus how men age. And that's true not just for the brain, but also when we think about things like frailty, right? Women become more frail with age than men. But when we think about age-related diseases, there's also a sex bias. Right, so for example, more men get Parkinson's disease, more men get type 2 diabetes than women, whereas there are more women living with Alzheimer's disease than men. And because we work on brain aging, we've been very curious about why that could be, what are the contributing factors. And there has been quite a lot of work on hormonal changes with age that occur in women due to menopause, but we wanted to actually look and see whether there's something actually going on within the cells with age. So not just systemically, but what about the brain cells, the neurons themselves with age? And so we did some deep sequencing of female neurons, and this was actually done in mice, but now we're looking in humans as well. And to our surprise, we found that there were changes on the X chromosome with age in the female brain that were not occurring in the male brain. And as you recall, females are XX in terms of their sex chromosome content at birth, whereas males are XY. It took us by surprise because what happens during development actually is early on, females will silence one of their X chromosomes, meaning they turn off all the gene expression on one of the X chromosomes, and that's so that we can have what's called dosage compensation. So that means that our one X chromosome is expressing the genes similar to what we have in males. And so we were surprised to learn that in females, there are actually major changes going on with age on the X chromosome that aren't occurring in males. This was specifically happening in neurons, and we picked it up in the hypothalamus, which is an important region of the brain for overall homeostasis, but it seems to be happening in other parts of the brain as well, like the hippocampus.
What does the hypothalamus do in us?
Yeah, great question. So the hypothalamus does a lot. I think of it as a very busy part of the brain. It's an evolutionarily ancient part of the brain, and it has a number of functions ranging from regulation of sleep and circadian rhythms, metabolic homeostasis with age, so that's our feeding, our energy expenditure, how much fat we store. It regulates reproduction, it regulates our pituitary gland, as well as a number of other functions. So again, very busy part of the brain. The functions of the the hypothalamus are actually critical for healthy aging, right? So sleep, we know, is dysregulated with age, and that's likely in part at least due to changes in a specific part of the hypothalamus called the suprachiasmatic nucleus.
Let's say if someone do develop some kind of difficulties with sleep and their rhythm of health, is there any treatment they could do at this stage?
I would say the more that you can learn about what's going on with your body, the better off you are. And so many people are data-driven. I'm a scientist, I'm data-driven. And so I think things like getting a glucose monitor, continuous glucose monitor, and actually measuring what are the glucose fluctuations and are there things that I can do to stabilize that can be very helpful. But I'm not promoting any particular product. But many people are actually tracking their sleep, and I think that's a great thing. People are finding out that actually if I have an extra glass of wine before I go to sleep, that's hugely disruptive for sleep patterns. I would encourage people to learn as much you can about your body and your patterns and what actually disrupts your sleep, what actually disrupts your or spikes your blood glucose. If you can get a handle on those things, then you can actually find interventions that work for you. And we're all different, right? The trackers that people are wearing are amazing, not just for individuals, but the fact that so many people are collecting this data, more is better, right? And that's true not just for tracking things like sleep, but there are data sets now that are out there that are, hundreds or potentially soon even thousands of individuals. And so we can actually look across populations, across individuals, and correlate, okay, who's doing really well in life and what are their biomarkers.
What drives you from very beginnings to become a neuroscientist? How you started all this?
Honestly, it was truly just that I loved being in the lab. I was the lab rat, right? Who, when I was an undergraduate, I just loved being in the lab. I loved doing experiments, and it was just such a new thing to move away from textbook science where you're sort of reading the facts or what you think are the facts, but not really thinking about the process of science. But when you go into the lab, that's true discovery, right? You're asking a question and you're designing an experiment, and at the end of the day, if you do it well, then you get an answer, and that's discovering something new for everyone. And so when I initially got into science, it was really just the process of discovery and actually being able to, make my own discoveries with my own hands that really got me excited.
What are that one particular moment to make you feel, okay, that's something I'm going to pursue?
I combined work that I had done when I was an undergraduate, which is actually where I first started working on aging, and I combined that with my graduate training, which was at the University of Washington, and there I started actually working on development and regeneration. And then when I moved to my postdoctoral work, which was at Stanford, I was just absolutely fascinated to learn that the brain can actually regenerate itself to some extent. So there is a region of the brain, very likely in humans as well, where new neurons are forming throughout life. And so the dogma has always been no new neurons, right? No new neurons are forming in the human brain in the adult. But it turns out that's very likely to be untrue. It's not happening everywhere in our brains, but it's happening in this specific region, which is the hippocampus, the region that's so important for learning and memory functions. And within the hippocampus, there's actually a very specific area, which is known as the dentate gyrus. And the dentate gyrus is an area that contains stem cells, neural stem cells. And it's these neural stem cells can actually make new neurons throughout life that integrate into the circuitry and are critical for forming new memories. One of the best activators is running.
Running.
Yeah. Well, so that's what mice love to do, right? And so mice love running. You give them a wheel, they just run and run and run. And so it has to be voluntary exercise. So if you stress, if you put an animal under stress, that it won't happen. The neurogenesis won't happen. But if you give a mouse a running wheel, it will run and run and run, and it will actually form new neurons in the hippocampus, even in an aged mouse, and you will actually get an increase in cognitive function. So in a cognitive task, the mouse will actually perform better. I think exercise, I would put that at the top of the list. It's not the only thing, though. Just simply staying active in terms of, brain activity. So basically giving mice new toys, letting them explore new things. And so I think that is going to translate to humans as well. And we know that brain function generally is improved through exercise and enrichment, but I think that the specific process of neurogenesis will also be improved in the human brain.
I really believe exercise have done a really, really great thing for my life.
Yeah, it's amazing, isn't it?
Yeah.
And so one thing you're doing is you're increasing blood flow to the brain, and that's a good thing. We do know that with age, there is decreased blood flow to the brain, and there is a vascular component to diseases like Alzheimer's disease. So vascular dementias are a huge problem. So increasing blood flow through exercise, it's not just anecdotal, it's a good thing.
So exercise can increase in blood flow. Is there anything else?
In functional MRI studies, which measure blood flow, you can actually see increases in the brain under certain tasks to different parts of the brain. Keeping your brain active either through learning new things or through social interactions, that's also a good way to stimulate the brain.
And what does stress do to our brain?
Yeah, so stress, unfortunately, is not good. You're probably not surprised to hear that. And neurogenesis is a great example of that. Stress will actually decrease new neuron formation in the hippocampus. A little bit of stress is okay. There is a process known as hormesis, where if we stress ourselves a little bit, that can actually stimulate some of these repair pathways that are actually good for aging. But chronic stress over and over on a daily basis is a bad thing. And there are actually links between things like social isolation and chronic stress, right? And that's a bad thing for neurogenesis as well as brain function more generally.
So how can we actually change our brain? Do you think as a human we are possible to reprogram our brain?
I think that's absolutely possible. And it's a matter of staying active, right? And sometimes our brain actually needs to forget. Mm-hmm. And so forgetting is actually not always a bad thing, right? We need to remember things in order to function. But in some cases, if we focus on a new thing and we practice that over and over again, we will strengthen those connections in the brain. We strengthen the synapses and the memories of that particular thing. And so there are ways to actually strengthen things, and then at the same time there will be some forgetting that will happen. And so I think of that as a form of retraining the brain for sure.
If we put our brain from zero when we just born till the day maybe we are going to pass away, could you walk us through of a life of brain development?
Yeah, so brain development is, a large part occurs before we're born, and then it continues. And in fact, the adolescent brain is still forming, and even into the early 20s, there's actually still a fair amount of neurogenesis, for example. And then between about 20 and till the end of life, that's when we see this decline in neurogenesis. And neurogenesis isn't the only form of plasticity that's occurring in the brain. Like we discussed, things like exercise, learning new tasks, that will actually change the connectivity in the brain as well. But that's more in the molecular connectivity between the neurons, right? Not necessarily the growth of new neurons. And that's something that is constant plasticity that's happening throughout our lives. And unfortunately, in the context of neurodegeneration, we lose that plasticity. So that is one reason why we start to struggle with cognitive function in the context of dementia.
I think when I read about it, I realized they said that when we are 2 years old, we probably have a lot of neurons, and then they start to decline. Could you tell us what is that about?
So one important point is that with aging, there isn't a lot of neuronal loss. So there isn't a lot of death of neurons with normal aging. And so I think this actually takes people by surprise, because we think, okay, with age we're probably losing neurons and they're dying, and we have more when we're young than when we're old. But actually the overall number is pretty stable in the adult human brain, but it's in individuals who have dementia, who have Alzheimer's disease, neurodegeneration. The feature of those diseases is the actual death of the neurons. And so what's happening to us, even starting right now, is we are losing our connectivity and some of our plasticity in our brains, but we actually don't have a lot of cell death. we do have inflammation that's coming up in our brains. So there's a lot that's changing, but not loss.
What will cause the inflammation in our brain?
In our brain, we actually have immune cells that are known as microglia. And the microglia are, just like our immune system more generally, they're our friends most of the time, but there are situations where they can become inflamed and become active, and they can actually cause some damage to neurons. And so what we see in Alzheimer's disease is that there's an inflamed state, an inflammatory state, where these microglia actually become activated. They change shape and they start secreting things, and the things that they are secreting are actually not necessarily good for neuronal function. So they're actually causing dysfunction in the brain in the context of disease.
What exactly microglia do in our brain?
With microglia, when they become activated, they secrete inflammatory cytokines, and those inflammatory molecules will actually act on the other cell types that are around, so the neurons, the oligodendrocytes, and will actually disrupt those cells, and they can no longer fire properly. They can no longer function normally in the context of these inflamed and activated microglia. And so that's hugely problematic. If we can find ways to decrease that inflammation, then that would be a good thing. There is some work that's being done looking at specific molecules, such as what's called the toll-like receptor, that is known to be an inflammatory molecule, and there's potential for targeting that receptor, but there isn't currently anything that's available, on the market in terms of a drug right now.
How early we can detect some kind of decline nowadays?
Yeah, so it's a good question, and that's actually one of the main challenges we have in the field is this long progression of disease that occurs, right? So people develop Alzheimer's disease over over decades, right? And so that makes it actually very difficult to understand the process. The good news is that the biomarkers are actually getting better. And so some of the tests, including a recent clinical test that came out, which is a blood test to pick up a phosphorylated form of tau early on in the disease, these things are changing the field rapidly to help us detect the disease early on. And that's what we really need right now. One of the main issues with Alzheimer's disease that I think takes people by surprise is that we actually don't really understand exactly what causes the disease. So we know that in individuals who have specific genetic mutations in genes such as presenilin, they can get an early onset form of the disease, and that is a defined cause of the disease. But that's not the Alzheimer's disease that most people get. Most people get the late onset version of the disease, and in most cases, we actually don't know what's going on. We know there are common features of the disease, like the buildup of amyloid plaques, the buildup of phosphorylated tau tangles, neuroinflammation, but we don't know what the first stage of the disease is and what the trigger is in most individuals.
So if someone already developed Alzheimer's, what are the current treatment for them?
So the treatments are limited right now, unfortunately. So that's the bad news. There are some drugs that will help with the symptoms of brain function in the disease. And then more recently, there has been development of drugs that target the amyloid plaques. And these drugs have actually been quite controversial. In some cases, they do seem to help. So these are drugs like lecanemab, aducanumab, and again, I'm not promoting any particular product, but there are some cases where those drugs seem to be effective. We know that they can clear the amyloid plaques in many individuals who are treated, but the issue is that in those individuals, even if their plaque situation improves and the plaques are cleared away, oftentimes there's no cognitive improvement. In some individuals, if you treat in the earlier stage of the disease and clear these plaques, you can get improvement. And so there are cases where it's certainly recommended, but in other people, unfortunately, it just hasn't really helped.
How we end up going to end our life, either decline of our physicality or decline of our brain.
I know, it's amazing. I find that amazing as well, that we see some individuals who are doing so well cognitively, and yet they have issues with mobility, right, or other things going on, and other people have the exact opposite situation, right? They can go out, they're active, and yet unfortunately they're developing disease. And so I totally agree. If we can figure out a way to sync this all up, then that would be an amazing thing.
So if we were to collecting our data to know what's going on in our mind, what are the current tests we can do to understand how healthy my brain is?
You can take a cognitive test, right? So you can actually do measurements of your performance, and that's really the best way to know what's going on in your brain, right? Because at the end of the day, that's what matters, right? So I would definitely start there. Part of the issue with brain imaging is that it can be somewhat crude. It's not as high resolution as we would like it to be, and so I think that's the future of brain science, will be to be able to tap into higher resolution imaging in the brain. But there's a lot that we can learn, for example, through the blood biomarkers, and so that's, for example, with Alzheimer's disease. In the past, the way to diagnosis was only to get a diagnosis was only through a brain scan, right? And you had to see the plaques in the brain, and that would be combined with cognitive function as well. But now we have blood tests to measure phospho-tau, and that's actually an early marker of the disease. So we can actually use the blood to understand what's going on in the brain. Unfortunately, we can't get in there with anything invasive. So the best proxies right now are cognitive function in performance and in the blood.
What's your point of view on brain-computer interface? What do you see the future of that going?
I think the future is amazing. I think it's going to help a lot of people. To me, watching what has developed in that field in the last decade or so, and the potential, I think that breakthroughs are coming very quickly in that area.
I know from your lab you do a lot of research and publish different papers. Is there any papers that your lab have published or you personally have published that you feel that's a very good piece of work?
We had a paper a couple of years ago where we made this discovery of changes on the X chromosome in females, and that is a paper that I'm very excited about, not just because of the initial finding that there's something going on in the X chromosome, but also just because it's opened up this whole new area of research for us that I did not actually anticipate that we would be going down this road, and that we would be, where we're at right now. One area that I think is very rapidly expanding in my lab, it's that area of X chromosome biology in the female brain.
What could that benefit us?
We're going to need some treatments that are more personalized treatments that are, going to help women or going to help men. It's not that we have a female bias. There are male biased diseases as well, but we need to gain an appreciation of how mechanisms of disease and mechanisms of aging generally are different, and what are the specific mechanisms where we can actually target in one sex where maybe it won't help others, and so we need to sort that out.
For the people who are peak performance CEOs, what is your suggestion for them to take off their brain health?
Sleep, eat well, exercise. Those are the main things. Social interaction. Get off your device and get out in the world and interact socially with individuals. Avoid loneliness. So I think that there's a lot that we can do just through improving our lifestyles.
If you think about future 10 years of neural health and brain health, where do you see us going?
I think the future is bright. I think the knowledge that we're gaining right now is going to help with brain aging, brain health, and like you said earlier, that is highly connected and integrated with our overall physical health. We're learning new things every day about, brain function, how to stay disease-free with age, and the future is bright, yeah.
What do you think is that one key habit that keep you going as who you are?
So I love to run, and I try to do strength training. I think that's particularly important for women, in fact, is to keep muscle mass up with age. It's like you were saying earlier, when you go from a desk to something physical, it's so stimulating and it sort of brings you back to life. And I feel that in my life as well. So if I'm starting to feel tired, if I have a little bit of a slump, don't go and lie down. Instead, I do something physical, and it can even be something short, and even a little bit can make a big difference. And so I would say, build physical activity as a habit in your daily life, and that's what I try to do to kind of keep everything going.
Is there any interesting thing coming out of your work in collaboration with other scientists here that you would love to share?
Absolutely. So I feel very fortunate to be here at the Buck Institute, and it's a really special place because everyone is interested in understanding the mechanisms of aging, and we're all committed to this mission of, how do we live better longer? So it's a unique place in the sense that pretty much everyone has multiple collaborations throughout the Buck, and I have a very exciting collaboration going on looking at quality control in the cell. So what I mean by that is, how do we get rid of damaged material in the cell? So that could be proteins, that could be organelles like mitochondria, and our cells actually have clearance systems that are great for getting rid of things when they're damaged, right? And we know that damage accumulates with age, and we also know that if we activate these clearance mechanisms, so these are things like the autophagy lysosome system, if we activate those systems, then we can actually get rid of the damage and improve cellular function even in the aged brain. And so this is a collaboration that I've been growing with a couple of different labs here at the Buck that I'm very excited about.
Thank you for today's time.
Thank you.
Thank you for your time, Ashley.
It was a lot of fun.
Thank you for watching the HumanWare project. To help us to getting more amazing content out there, please subscribe. It means a lot and it will help us to grow. And let's go on this journey together.